Mm. Fix. Right. So we learn here there are two factors which can increase or which can decrease the rate of reaction. Two factors which can increase or decrease rate of reaction when enzymes are involved. Second one, pH. Right. I think most of them they don't know what is the meaning of pH. The pH stand for potential of hydrogen ions. So P and H, we write pH, potential of hydrogen ions. Okay. Uh, left. So again, we try to draw this graph. So X is independent variable, Y is dependent variable, and this is rate of reaction. And that's a pH. So same graph like before, rate of reaction, dependent variable, we measure the rate of reaction and we try to measure rate of reaction at each pH. So we have a different pH. So lowest pH is 1, the highest pH is 14. One and 14. So chemically, what's actually happening when you go this direction, there are more hydrogen ions formed. The hydrogen ions are very strong. They can make so much of chemical reactions. So when you go these directions, the more OH ions formed, negative OH ion formed, and they're also very strong. So then enzyme reactions, again, it goes like this and then decrease. So pH, which is the best rate of reaction, is called optimum pH. Optimum pH. Now, why rate is low, why rate is high, we can learn. So can you again copy this graph to your notes? So why this and why this? So again, we have two questions. Why rate is low at high pH? Why rate is low at high pH and low pH? Right, again, you can copy this graph and then you can send me a message copied. One, done. Actually, Synergia. Okay, lithium also completed. Right, two more to complete. Done, done. Okay. I think some of them completed. We wait few seconds. Others can also complete. Shumai also done. It's another one. Ashwin, right? Ashwin. Ashwini also done. Right, I'm going to give the answer for both question as a one answer. So answer is enzyme denature. So enzymes denature at both high pH and low pH, enzyme denature. So what's the meaning of denature? Denature is like this. You can understand uh, answer for C and D, right? This is answer for C and D. Answer for C and D. C and D for both. We have one answer because both in enzyme denature. Here denature and here also denature. So what is the meaning of denature? You can understand proteins. Proteins are very sensitive for heat. you can see this one. If you fry egg, if you boil egg, the eggs, mostly the white color, white region is a protein. The white color part of the egg is protein. So then the protein completely change, it gets solid. You can't bring them back again. If you boil the egg, the white color proteins become solid. You cannot bring back to liquid again. If you fry, the white color become again solid. You can't bring back to liquid. So it's completely change the protein with the pH. Enzyme denature, denature is shape change. So active site shape change. The meaning of denature is active site shape change. The meaning of denature is active site shape change. At low pH, enzyme denature. At high pH, enzyme denature. The meaning of denature is shape change, the enzyme shape change, active site shape change. Answer for this question is denature. For both denature. pH change the shape of enzyme. pH change the shape of proteins. Okay, now the last thing we're going to learn, what is a buffer. So usually in chemical reaction, pH changes over the time. So usually in chemical reaction, pH changes over the time. So it is not the pH, beginning of the pH is not the same as end of the pH. So now important thing, if we add buffer, it prevent the change of pH. Other word, it keeps pH constant. So what is a buffer? Buffer is a solution. It keeps pH constant. In a chemical reaction, it doesn't allow to change the pH. So that's the meaning of buffer. So if we add buffer, if we add buffer, it prevents the change of pH. It keeps pH constant. Right, you can also copy this down. You can write now what is a buffer solution. What is buffer solution? Usually in chemical reactions, pH changes over the time. If we add buffer, it prevents the change of pH. It keeps pH constant. Usually in chemical reactions, pH changes over the time. If we add buffer, it prevents the change of pH. It keeps pH constant. Right, we'll come back again to this place, and you can complete this. So we almost revise enzymes, the most important thing about enzymes, but still we have one question to answer. Temperature, what is happening at low temperature? What is happening at high temperature? Why rate is low in both temperatures? Right, so I think we go back to the question again. Mix 5 cm cube of pineapple juice with buffer pH 4 buffer. pH 4 buffer. Pineapple juice has enzyme. That's a protease enzyme. Place 1 cm of pineapple juice and buffer solution on top of the gelatin. Now we're going to add the buffer solution and the pineapple juice onto the top. Leave for 1 hour in water bath. So water bath is the instrument we set temperature constant. So that's a constant temperature. So this comes under control variable. This is a control variable. We have to keep temperature constant because temperature can affect rate of reaction. If temperature change, rate of reaction change. So we have to keep it constant. Then has no effect on temperature. Now what will happen when you keep 1 hour? The enzyme start to break down this gelatin. So enzymes start to break down the gelatin, and over the time, the height of the gelatin decrease. Now you see the height is this much after 1 hour because enzymes break down gelatin. Protease is enzyme, gelatin is a protein. Repeat the method 3 more times. The diagram shows part of the student method. Pineapple juice and pH buffer solution, gelatin protein. Okay, next. Right now, I will explain your answer for this with the 3 marks. The table shows the student result for the volume of gelatin digested at pH 4. So pH 4 is a constant because buffer solution, as I mentioned, keep pH constant, pH not change, pH is always 4. Temperature is 37, pH is 4. They have four tubes, one, two, three, four. Then the volume of gelatin digested in centimeter is given. Right now, I will give the answer, but I will give a chance for you to calculate mean volume of gelatin digested in cm cube. Give your answer to two decimal places. So it is already given in a cm cube. So I will give it two decimal place. I will give you a chance. First, you calculate and send me the answer through chat. You can use your calculator to calculate the mean value. Usually, I don't want to explain how to calculate mean value. You have been practicing this from class 8, class 9, and now in the maths anyway, you are doing this, how to calculate mean value. Okay, I write the answers of your all answers which I get. Others can also send you an answer. This is one answer I got. So far there's only one answer came. Hmm. Okay, I think I don't get any answers. Seems to be a problem that you don't know maybe how to calculate mean value. So you can use calculators, then it's faster. Okay, I think you are in trouble. Right, I will show you how to calculate mean value for any example, right? So I will show you example. What is the mean value? So mean value. So mean value is the value which going to represent the group. For example, if there is a several values in this group, let's say 6 is a one value and 4 is another value and 10 is another value and 5 is another value and uh, 20 is another value. So this is a one value 6, 5, 10, 20 and 4. So I have 5 values in this group now. So I want to get only one value from this group as a rep representing value. So this is called representing value. Representing value. Six, 5, 4, 10, and 20. But I need only one value to here to show what is the best value to represent all these values. So 4, 6, 10, 5, and 20. I cannot take 4, 4 is too low. I cannot take 20, 20 is too high. But I can think about take some values like 10 because in between 20 and 4. But I can't do like that. There's a mathematical method to do that. The method is number one, add all the numbers. First, you add all the numbers. Then, second, divide by number of uh, data. So this is a one data, 6 is a one data, 10 is a one data, 4 is a one data, 5 is a one data, 20 is one data. So 1, 2, 3, 4, 5. So it is 5. I have 5 numbers. Then the mean is you have to add 6 plus 10 plus 5 plus 20 plus 4 divide by 5. This is a mean value. So you have to add all these numbers now. So when you add all these numbers, 6 plus 4 is 10, 10 plus 10 is 20, 20 plus 20 is 40, 40 plus 5 is 45. The total is 45 divided by 5. This is 9. The mean value is 9. Now I can write here mean value is 9. So this is the mean value. So this is the mean value. Right now, if you understand how to calculate mean value quickly, calculate the mean value for these numbers. So you have to calculate, these are the numbers. Volume of gelatin digested, and here the numbers. Tube number 1, volume is this, tube number this, tube number 3, tube number 4. So we have to get the mean value for this. What is the mean value for this number? Another answer is 0.89. So I got another answer 0.89, 0.90. So if you add this number, total is 3.59. So 3.59, you have to divide by 1, 2, 3, 4 numbers. 3.59 you have to divide by 4. So 936. Yes, I got 0.89. So 0.89 is the answer I got. And 3, then I get 30. 30 is 7. So it's a 28. So I did manually. You can use the computer, calculators. So I get the answer 8, 9. 897. So my answer is 897. You see, my answer is 0.897. Now they ask, give your answer to 2 decimal places. So I have here 3 decimal place. So I want to give 2 decimal place. So now 7 is bigger than 5. So 7 is bigger than 5. So I have to round up now. I'm going to make this round to 10. So the 9 become 10. So when 9 become 10, this become 9. So 0.90. So the most correct answer is 0.90. 0.90 after we rounded up to 2 decimal place. That's called rounding the numbers to 2 decimal place. This is your first lesson in your maths. If you take your maths book, the rounding numbers is the first lessons. So most of them end up with 8, 9 mean it is 897. So 897 round up is a 0.90. Okay. But there is something you have to learn. We all are wrong. All are wrong. This is not the correct value. You don't get the marks. So this is 3 marks question. Do you think they will give 3 mark just to calculate this mean value? In your maths paper, you get 1 mark. To just this kind of a math, you get a 1 maximum 2.